Hi, everyone. Welcome to the Youth Advisory Council interview series. We, the Youth Advisory Council at the Center for Youth Bipolar Disorder, CYBD, at CAMH, have started the series to try to raise awareness and translate knowledge from the ongoing bipolar disorder research in a way that is accessible and meaningful to patients and families. This is the first interview in the series. CYBD's Youth Advisory Council, also called the YAC, was established in 2021 and is composed of 6 youth advisors with bipolar disorder. The YAC team allows the opportunity to meaningfully engage with our patients and integrate their voices towards education, stigma reduction, and advocacy efforts in the field of youth bipolar disorder. A great accomplishment of the YAC has been the creation of Never Alone, a Youth's Guide to Bipolar Disorder, which is an informative and inspirational booklet that shares first-hand narratives of youth with bipolar disorder, written by youth for youth. This series is an extension of the YAC's team's efforts. Today, we have 3 youth advisors interviewing one of the CYBD researchers. My name is Kaylee, and this is my second year on the Youth Advisory Committee. I'm a second-year PhD student at U of T.
My name is Nick, and this is my first year on the Youth Advisory Committee. I am going into my second year of an undergraduate degree in concurrent education.
My name is Tara, and this is my second term on the Youth Advisory Committee. I'm a medical student heading into third year.
Today, we have Joyce Jiang here, who is a PhD student at CYBD. She's here to talk to us about her research. Thanks for coming, Joyce.
Thank you for having me.
So Joyce, we just wanted to start off by asking a little bit about what your current role at the CYBD is, what it entails, and how long you've been here for.
Yeah, so I'm currently a PhD student at CYBD. My work mainly involves first finishing my thesis, so writing the different papers for different projects, but also it involves a lot of other teamwork, so such as running clinical study visits or writing grants, and also involves a lot of data set cleaning or imaging QC that requires a lot of teamwork. I've been at CYBD for 4 years now, so I did a 1-year undergraduate research projects at CYBD, and after that, I did 3 years of PhD, so I'm currently entering my 4th year of PhD.
Thanks so much, Joyce. Could you please give us a brief summary on your educational and work background that led you here?
Sure. So I did my undergrad also in the Department of Pharmacology and Toxicology at U of T. So in my first year, I took a Introduction to Psychology course, and that got me really interested in psychiatric disorders. So in my second year of undergrad, I took a psychology minor. Then after a year, I realized I wasn't really interested into psychology, but rather I'm more interested in psychiatry specifically. So I started to look for different research opportunities focusing on psychiatric disorders. I started by doing some cell works and animal works in wet labs, so which requires actually running the experiments like pipetting and stuff, cell culturing. And then I got more interested into something. I wanted to do something that's more clinically relevant, because the cell works and animal works are really more on the basic research side. So sometimes it's very easy to get lost and can't really figure out what's the clinical relevancy of the research I was working on. So in my first year, I joined Dr. Benjamin Goldstein's lab to focus on clinical research, and I really enjoyed it because I really enjoyed the lab. So after that, I just continued on to do a PhD in the same lab as CYBD.
Thank you. That is a really, really cool story on how you got here. Could you tell us a little bit more about what your current body of work is focused on? How did you decide to pursue this specific topic of research?
Yeah, so my current work mainly focuses on molecular genetics in relation to some brain structural phenotypes, brain function, cognition, and clinical characteristics in youth with bipolar disorder. So the specific molecular genetics that I focus on is called polygenic risk score, which is basically a score to estimate individuals' genetic susceptibility to a condition. So for example, for bipolar disorder, we can generate polygenic risk scores for bipolar disorder. And if an individual has a higher score, that suggests a higher genetic risk for bipolar disorder. I decided to pursue this topic of research because, like I mentioned earlier, I was really interested in something that's more clinically relevant. So this polygenic risk score, although it's still in its infancy, but it has potential to become a really useful clinical tool to help validate clinical diagnoses or stratify risk, or just generally help us to understand youth bipolar disorder more.
Thank you. We'd love to hear about your research a bit more in depth. We were hoping that you could break it down for us and perhaps explain it at the level of a middle school student. So we were interested in your paper titled Association of Lithium and Second Generation Antipsychotics with Neurocognition in Youth with Bipolar Disorder. Yes, we'd love if we could hear a brief synopsis of that.
Yeah, so this paper, I did this paper during my first year of undergrad. So when I was doing that undergrad research project at the lab. So this paper specifically looks at whether the current use of lithium or second generation antipsychotics was associated with neurocognitive performance in youth with bipolar disorder. So we decided to look at this topic because lithium and second generation antipsychotics are two frontline anti-manic medications for youth with bipolar disorder. And it is one common belief about these medications is that they can cause neurocognitive decline or impairments, which in turn leads to non-adherence to the medications. However, there are only very few prior studies that examine this topic, and the sample size of these prior studies are relatively smaller. So we decided to look at this topic again, but in a much larger sample size. And for this study, we examined different aspects of neurocognition, including sustained attention, cognitive flexibility, emotional processing, and spatial planning. I will explain more how these are measured later. But we found that the current use of lithium and second generation antipsychotics was not associated with neurocognitive performance in these aspects, in these tests. We were, at least statistically, the association was not significant, which may suggest that these leading mood stabilizing medications are not associated with significant neurocognitive impairments in youth with bipolar disorder. Yeah, that's basically brief overview of that paper.
That's very interesting and very cool. Thank you for sharing. I wanted to circle back and talk a little bit about neurocognition that you mentioned. So what exactly is neurocognition and how did you use it in your study? How was it measured?
Yeah, so neurocognition is a very general term. In a more simple way, it's how our brain handle different daily tasks. So for example, in a class and you try to focus on the lecture, this is a time when your brain is when you need sustained attention, which is a sub aspects of neurocognition, is a subdomain of neurocognition, and there are other tasks that you need to do in your daily life. So, for example, you're solving different problems, which will require cognitive flexibility. So you really need to be flexible on how you solve different tasks like math or physics. You need to be able to swift between different tasks. And there are other cases, another aspect another domain that we tested was emotional processing. So when you're talking to someone and you try to interpret it, whether that person is sad or happy, what that person's face look like, is that a happy face, is that a sad face? That's when your brain is involved to figure out that to do the emotional processing. So basically, neurocognition is how we handle different tasks in our daily life, which will involve in different aspects of different domains of neurocognition. So it was measured. We measured neurocognition using a toolbox called Cambridge Neuropsychological Tests Automated Battery, which is basically a battery of tasks that requires a touch screen and high sensitivity trigger buttons to record responses to each individual tasks. So for this study, we examined 4 neurocognitive domains, including cognitive flexibility, which I mentioned earlier is something we need when we are trying to adapt to different tasks or different tasks. And sustained attention, which is required when we try to focus on our specific things such as lecture, and spatial planning, which is required when we're, for example, when we're organizing our furnitures or organizing a painting. And finally, emotional processing, which is required when we're try to understand other people's emotion, whether they're happy or sad or neutral. So these domains were picked because they were commonly found to be impaired among individuals with bipolar disorders. And for each domain, there are several tests for the participants to complete. And when we then use these scores from all individual tests to generate a composite or overall score for each specific domain. So we can get a score for cognitive flexibility, a score for sustained attention, and so on separately for each task, each domain. Yeah.
Thank you,
Hope
Joyce.
that's clear.
So you mentioned that your research showed no statistically significant association. Do you think there's any major limitations to your study?
Yeah, so the first thing is this was a observational and cross-sectional study, meaning that we did not follow participants over time. We just examined their neurocognitive performance at this one time point. And therefore, we cannot establish the directionality of observed association between neurocognitive performance and current lithium or second generation antipsychotics use, which means that we cannot be sure, for example, whether current use of these medication cause neurocognitive impairments, or in turn, if neurocognitive impairments may lead to more use of these medications. So we cannot be sure about this directionality. And also, we did not systematically collect data on the duration of treatment or the dosage of these medications or the adherence to treatment. And all these factors could impact the observed associations between medication and neurocognitive performance, because these are some important factors that could impact the neurocognitive performance. So that's another major limitations. So definitely in the future, we need randomized control clinical trials, which can test the effect of these medications in a more controlled environment and can compare these medications to placebos, for example. And these kind of trials will help us to understand the more definite effects of these antimanic medications on neurocognitive performance in youth with bipolar disorder.
Thank you. You alluded a little bit to what I'm going to ask next, but just to make it really clear, how do you think your research is particularly relevant for patients, either now or in the future? And in other words, how can the findings be applied to their lives?
Yeah, so specifically for that paper that we were just talking about, I think the main relevance is that hopefully it helps solve doubts over these antiemetic medications that, at least in our sample, these medications were not associated with significant neurocognitive impairments, which hopefully can improve adherence to these medications in youth with bipolar disorder. However, because of the limitations that I mentioned earlier, I'd say clinically, we can't just rely on this one paper, right? But at least this is some promising evidence. We definitely need randomized control trials in the future to be more certain. But at least this is some promising evidence that antimanic medications are not associated with significant neurocognitive impairments. In regards to my overall thesis work, so my thesis, as I mentioned earlier, focuses on polygenic risk scores. And I also mentioned that this is still in its infancy, so polygenic risk score alone cannot be used in a clinical setting. But there is one prior study that combined this polygenic risk score with other traditional risk factors, and together create this risk calculator that has a 80% accuracy in predicting youth with bipolar disorder versus youth without bipolar disorder. So I'd say at the current stage, polygenic risk scores probably cannot be applied in a clinical setting yet, but let's say in the future, I believe it has a great potential to be used as a tool to validate diagnoses, to predict risk, and most important, to validate the diagnosis of bipolar disorder in youth, because one major stigma over youth bipolar disorder is that it does not exist. A lot of people believe that bipolar disorder only exists in adults, but cannot exist in youth. So our findings, our finding on the molecular genetics is really a very objective evidence that shows that our clinical diagnoses can be supported by this objective biological evidence.
Thank you. So you mentioned that this is something that's going to take a lot of research in the future. Is this something that you plan on looking into specifically? And is there any other questions that arose as a result of this research?
Yeah, so like I said, the polygenic risk score is still in its infancy for probably two reasons. First is when polygenic risk score for bipolar disorder is examined in either youth sample or adult sample, we found that it has very little explanatory power, which means that it only contribute to a very small proportion of the overall risk for bipolar disorder. Basically means that polygenic risk score is not the only thing that could lead to increased risk for bipolar disorder. There's a lot of other risk factors that are not considered into this score. And another thing is that currently polygenic risk score can only be applied to individuals of European ancestry, just because a lot of prior genetic studies were conducted among adults with adults of European ancestry. So that's just unfortunate that a lot of genetic findings, either specific to polygenic risk scores or other or more general genetic findings, can only be applied to individuals of European ancestry. However, there is currently and also in the future, there's a lot of global efforts that are developing multi-ancestry polygenic risk scores and other statistical methods to hopefully improve the generalizability of these genetic findings to individuals of non-European ancestry. So that's definitely something I'm looking forward to look at in the future, to generate multi-ancestry polygenic risk scores and not just limit our sample to youth of European ancestry, but to include a bigger sample with more diverse ethnicity background.
That's really cool and very relevant. Thank you. Why is it important to have youth with lived experience involved in the research process?
Yeah, I think it is very important in probably two aspects of during the research process. The first is when we are designing the research question. So there's a lot of questions that can be asked, a lot of studies that can be done, but the funding is very limited. So we really need to pick our priority and ask what's the really clinically important question to study, what's a really important topic to focus on. And that's when youth with lived experience can be very, very important to help us pin down that one question that will be really clinically significant. And also, the second aspect is when we are designing the whole study procedure, the study protocol. So a lot of our study procedures involve surveys and questionnaires. So we really want to make sure that our participants feel comfortable with the entire study visit and also comfortable with that surveys and questionnaires. That's when we can get we can consult with youth with lived experience to go over the study procedure and to see if it makes sense, if it's comfortable, if there's anything triggering. I think that's when it's very important to have youth with lived experience. Yeah.
Thank you. We completely agree that not only is it so important for youth to be involved, but it can also be so empowering. I know that's something that the three of us have really felt during our time on the Youth Advisory Council. So thank you for sharing that. Joyce, we were wondering, when you're considering next steps for your research, what are you most excited and hopeful about and why?
Yeah, I briefly mentioned this earlier, but my research on the polygenic risk score that I'm studying currently only have very limited power, explanatory power, or power in predicting youth with bipolar disorder versus youth without bipolar disorder, just because it's a very small contributing factor. There's a lot of other factors that we are not considering, such as environmental risk factors or other rare genetic factors that are not calculated into this polygenic risk score. So in the future, I'm definitely excited to considering all those other factors and try to come up with a model that integrates all these different pieces together to hopefully get a better prediction model. I've seen people doing that using AI. So they feed the AI, they feed all the neural imaging scans into the AI, and AI can achieve almost 90% prediction accuracy. So it will be interesting to also feed polygenic risk scores into that AI model. But I'm not a data scientist, but I'm looking forward to hopefully have some collaborations with some other data scientists.
Thank you, Joyce. So it's been great so far to hear your answers to our questions, but is there anything else that you were hoping to share about your research or your journey specifically?
Yeah, so about my my journey at CYBD, I say I just really enjoyed the lab environment, not just environment, but also the research environment, because my work focused on genetics, but another big aspect of our research also focus on cardiovascular risk in relation to youth youth bipolar disorder. So I got a lot of opportunities to be exposed to other research that I'm not actually focusing on for my thesis. So I got exposed to a lot of diverse research topics, not just to focus on the niche of genetic world. I guess that's a lot of that's normally how a PhD is. You focus so deep on one niche topic, but never got chance to be exposed to other things. But at CYBD, because the research we're doing is so diverse and so broad, I got a lot of different opportunities and experience to learn other things as well. That's something that I just really enjoyed during my journey of research.
Wow, thank you so much for your time and for sharing your knowledge and your insight. We learned a lot about polygenic risk score and why youth with lived experience should get involved in research, and why the research process should include people with lived experience. And we know our audience will appreciate everything that you shared with us today. Thank you so much. And to our listeners, that's all the time we have for this interview, but stay tuned for our next interview in the series. Thank you.